Hello, my name is Shanab Daniel Shenbigan, matrix number 200350, and I'm part of the Bachelor of Science in Biotechnology of Honors, first year. In this video, I will be presenting on two experimental procedures. Firstly will be the streaking plate technique, and second would be the gram staining technique. So let's get right to it. So let's talk a bit about the principle behind this experiment. So it actually has to do with the isolation of discrete colonies from a mixed culture, in which you will actually isolate discrete colonies, usually in a mixed culture, in order to obtain a pure culture, which is just it contains only one type of organism, and you can actually use this to study their biochemical or morphological properties. So in the terms of a streak plate technique, what is going to be done, it's actually considered a dilutive method, in which you are spreading a loop full of culture over the surface in 4 different quadrants, and in each subsequent quadrant the microorganisms will be separated further and further apart from each other, and by the time you reach the last quadrant, it will actually be individual cells will actually be far apart enough for you to separate them from each other. And this is very useful in terms of separating different species from one another, especially if it is done in a mixture of species. So now we're going to get into the procedure. Before we get into how to do the experiment, we're going to talk about the materials first. So we have our inoculating loop, we have our petri dish complete with agar, if you can examine it here. We have our container, which will take the place of a bacterial culture container, and we have our Bunsen burner, which will be replaced with a cup for safety precautions. So firstly, the most important thing is always aseptic technique. So we're going to heat up our loop until it's cool, until it's red. The loop turns red. Then we are going to let it cool down. Now after this happens, we are obviously going to dip it into the culture. This is obviously replaced with a container, a culture, and it is taken out. And the top of the culture is also, of the container of the culture is also heated. So now we already have a loop full of bacteria. We have our petri dish. So right now we're going to actually separate it into 4 quadrants. We'll imagine 4 quadrants. So our first one, we're going to slowly streak it across the surface. Let's imagine it's the first quadrant. We're going to streak it across the surface. Right now that is done, we're going to heat this up again, the loop again until it's red. Give it a bit of time to cool down. And now we're going to imagine this is the second quadrant. We're going to also spread the same loop from the end of the first quadrant, but not touching the first quadrant. Now that one's done. Now for the third part, we're going to also heat it up again, very important. Let it cool down, and now we turn it this way 90 degrees, and then we spread the third on the third quadrant in the exact same method, starting from the end of the second quadrant but not mixing with the second quadrant. Now we have our fourth and last quadrant. We don't have to flame the loop, and this is because the cells are already spread out enough from our flaming of the first and second and third quadrants. So for the fourth we won't have to, so we just do the exact same thing. So yes, that will be the procedure. So we will actually be streaking our loop against 4 different quadrants. So the expected results in a perfect situation for this experiment would actually have 4 particular patterns of growth on your petri dish, which corresponds to the 4 quadrants that were mentioned just now. So the first one would be heavy confluent growth, because during your first method of streaking, you are having a lot of bacteria in your loop. So a lot of bacteria will grow on the agar plate, followed with heavy growth, which will be slightly less, which is on account of the heating process, followed with light growth, which is also due to the heating, and then followed by discrete colonies, which will be in your 4th quadrant. So this will be the perfect scenario and probably the expected result. So now we're going to go into our second experiment, which is gram staining techniques. So gram staining is actually known as a differential staining, and this is because it is able to differentiate between 2 types of bacteria, namely the gram-positive and gram-negative bacteria. 4 stains are incorporated in this particular technique, which is the primary stain, crystal violet, mordant, which is grams iodine, decolorizing agent, which is ethyl alcohol, and the counter stain, which is safranin. So this particular type of staining is actually used to differentiate, as I mentioned, gram-negative and gram-positive bacteria, which have different morphological characteristics, in which gram-negative bacteria have a thin peptidoglycan layer, so they can be counterstained easily by safranin, whereas gram-positive bacteria have a very thick peptidoglycan layer, so they are not easily counterstained by any type of stain. So basically, based on this knowledge, we will be able to carry out this experiment and figure out whether we can distinguish gram-positive and gram-negative bacteria. So let's get right into it. So let's talk a bit about the materials that are used in the experiment. So we have our inoculating loop, our Bunsen burner, our bacterial culture. We have our basin with holders here, which will be used to place the slide on top later. We will cover it later. Next, we have our slide. We have our stains. So we couldn't obtain droppers, so we'll be using these particular containers as substitutes. We have first our crystal violet, which is our primary stain. Next, we have our mordant, which is iodine. We have our decolorizing agent, which is alcohol, ethyl alcohol. And last, we have our counter stain, which is safranin. So now, let's get right into it. Our first step is to gently flood the smear with our crystal violet. And we'll let it stand for 1 minute. So let's do that right now. So the smear, we'll just gently flood it. We'll leave this for a minute. So let's give it a minute. And we'll talk a bit about why this crystal violet is important. So it acts as a primary stain, which actually works to stain the components of the cell. So let's just give it a minute first. Okay, it looks like a minute has passed. So now, right now, we're going to gently wash the smear with tap water. So I'll be using this spray as water to substitute water. Alright, perfect. So now we can see that it's been removed. Now on to our second step. We're going to use our mordant, which is iodine, which actually works to amplify this color of the primary stain. So our mordant, which is iodine in this case, we'll just be putting it here. Alright. So just pour it gently, and we're going to also leave this for a minute. Okay, a minute has passed. I'm going to do the same step. We're going to gently wash it off with tap water. Okay, that's done. It is removed off the slide. Alright, now we're going to do the decolorizing step. Now the decolorizing step is actually done by a decolorizing agent. In this case, it's ethyl alcohol. And its reason that we are using it is so that we can remove the primary stain. Now this may seem counterintuitive, but it's actually so that we will be able to counter stain it with this particular color in the next step. And this means that if this manages to counter stain the cell's components, that from the primary stain, those particular components can be restained or counter stained, more accurately, by this particular safranin. So we're going to just gently just pour in a few drops because we don't want to over decolorize it. So just a few drops, and that should be enough because the alcohol is already run clear. So we'll leave it here, and now we're going to just gently wash it off with tap water. Okay, looks like that's good. Now on to our last step, which is the counter staining step, which we'll be using safranin. Safranin works to counter stain any of the components that were decolorized by the alcohol just now. So it means that any particular decolorized components will exhibit a pink stain instead of a purple stain because they have already been decolorized. So now we're going to just pour a few drops now, and we're going to do it for 45 seconds. Okay, now that the counter staining is done, we're going to gently wash it off with tap water. Okay, looks like the staining process is done. Now that the staining is done, the gram staining is done, we are going to place it onto bibulous paper, and we're going to blot it dry gently so that we do not bother the smear. So we're going to gently do it. And next, we will be observing it under a microscope. So that's it for the procedure. So as you mentioned before, there will be two types of bacteria that will be demonstrated or examined in this experiment: gram-positive and gram-negative bacteria. The gram-positive bacteria will actually be stained purple because they have a thick peptidoglycan layer, so they're not easily decolorized. Whereas the thinner peptidoglycan layer in the gram-negative bacteria will actually exhibit a pink stain from the safranin because they are easily counter-stained. So these are the results that we will be seeing in this experiment. I hope that this is sufficient, and thank you very much for watching.